today's focus is gene knockout via CRISPR. So if you're interested in other applications or the general information about the CRISPR technology, so Shuang also mentioned that we have a series of videos and recorded webinars. You can find them on YouTube or on OriGene website. Okay, so here is the overview. First, I will give you a brief introduction about CRISPR-Cas9 system and the main applications, what you can do with the CRISPR technology. And then I will talk about the OriGene's CRISPR gene knockout kit, follows with a step-by-step protocol. And then I will give you some validation data. So everybody knew that CRISPR-Cas9 is groundbreaking in genome editing. It is very simple and efficient. It's an RNA-guided genome editing tool. It needs two components, Cas9, the nucleus, which cuts the DNA both strands, and the guide RNA, gRNA, which brings Cas9 to the location sequence specifically. So this is the diagram. Here, that's the chromosome. The gray blob, that's the Cas9, which cuts the DNA both strands, leading to double-stranded break. This is the gRNA. And the green part, that's the target-specific sequence, 20 base pairs. And the blue part, that's the gRNA scaffold, which does not change and can be built in a vector. So after the double-stranded break, how is genome editing achieved? Genome editing is achieved by DNA repair mechanism. There are two main repair mechanisms. One is NHEJ, non-homologous end joining. In this repair, there's no repair template. So the ends of the bricks are joined together, and this process is error-prone, which you will get variable mutations, insertions, deletions. So if the insertions, deletions change the protein coding frame, you will get gene knockout. And another repair is HDR, homology-directed repair. So in this repair, you will need a donor template. It has homologous sequences. The desired change is in the middle. So through homologous recombination, the desired change will be integrated into the genome. So this way, you will get precise, by your design, gene knockout, specific mutations, SNP, deletions, insertions, and gene tagging. You can also knock in a reporter gene for your promoter study. So this shows the main applications using CRISPR-Cas9. So some people need to make specific gene mutations in the genome, either insertions or deletions, such as SNPs. Genome editing is working on genome level, so you need to use the genomic sequence to design gRNAs. So the diagram here shows this is promoter. The boxes are exons. The lines are introns. So you need to design, use genomic sequence to design gRNAs around the mutation region, and the gRNA will cut at the area position, and the mutation sequence is provided on the donor DNA. So another application is gene tagging. You can tag at the N-terminus or C-terminus. So not every gene has good antibody, so that's why the gene tagging is very useful to study your gene function. The diagram here shows the GFP tagging at the C-terminus. So you design gRNA targeting around the C-terminus of the gene. And on the donor DNA, the left arm is the C-terminus of the ORF without the stop codon, follows with the GFP. And then the right arm will be the stop codon follows with the 3' UTR. So this way, through homologous recombination, the GFP will be inserted right downstream of the C-terminus. As Sean mentioned earlier, that Another important application is gene knockout. So if you only have CRISPR-Cas9, it will cut the genome and you will have indels. You will have variable insertion deletions. If the insertion deletions change the protein reading frame, you will get gene knockout. And another way is use homologous recombination. On the donor DNA, you also have a reporter selection cassette to replace the target gene. So this way, you will have you can use antibiotics to help selecting your edited cells. And this is the main focus of today's presentation. So next, I will talk about OriGene's CRISPR gene knockout kit. Before going to the details, I want to show you CRISPR's offering from OriGene. This is the CRISPR landing page. There are a couple of tabs on the top, and you will gene knockout kit or CRISPR vectors, germ cloning and donor construction, AAVS1 transgene insertion, and the CRISPR starter kit. So the CRISPR we have a variety of different CRISPR vectors. This is all-in-one CRISPR vectors because it contains both the gRNA and the Cas9, so it will lead to double-stranded break. And this group, that's the T7 vectors. You can make a gRNA and a Cas9 mRNA in vitro using T7 transcription kit. And we also offer nakeds. For the gRNA cloning and donor construction, we offer custom gRNA cloning and donor construction. So we have a free gRNA design tool. So you can design your target sequence and just put in the genomic sequence. So if you can give us the target sequence and we can clone into any of our CRISPR vectors. If you don't want to do design, you want to leave to the CRISPR experts, we can do the design and cloning into the CRISPR vector too under the gRNA cloning service. If you need to do homologous recombination, we can offer donor vector construction because OriGene has a gene synthesis arm, Blue Hammer, so we can provide any sequence of your interest. So the AAVS1 transgene insertion, that's for transgene exogenous gene insertion into human genome at a specific location, AAVS1 site. That's a safe location, and the expression is high if you want to express an exogenous gene in cell. So this can be done via CRISPR. The CRISPR-Cas starter kit, that's the positive control to test your CRISPR system in your cells. This is a kit HA tagging. So now we are going to talk about the CRISPR gene knockout kit. So it's ready to use, and you don't have to do any gRNA design and cloning yourself. When you receive it, it's ready to do your experiments. So it's a complete kit. It has components, 2 gRNA vectors, and 1 donor vector, 1 control. So you can do gene knockout, and in the meantime, knock in the GFP payload cassette to help selecting your cells. The 2 gRNA vectors are cloned in the pCas9 vector, one of the all-in-one vectors. So in this vector, the gRNA will be expressed under U6 promoter, and Cas9 is under CMV promoter. So this way, it will lead to targeted double-stranded break. The 2 gRNAs target similar location. They are provided individually, 2 individual tubes. They also should be tested separately to ensure efficient cleavage. 1 GFP payload donor vector, of course, with the homologous arms already cloned in it. That's the left homologous arm, right homologous arm, GFP payload cassette. Pyromycin marker is under PGK promoter, and this selection are flagged by LuxP. So after integration, if you want to remove the pure selection, you can use Cray to flux out. The scramble control is also provided in pCas9 vector, serve as a negative control. So how does the kit work and how is it designed? So when you do a co-transfection of the gRNA and donor vector into your cells, So the gRNA and the Cas9 will lead to targeted double-stranded break, and the donor vector has homologous sequences. Through a homologous recombination, the GFP puro will replace your target gene, so that gene is knocked out. So how it's designed? To do gene knockout, the targeting sites should be around the starter codon region, ATG. And on the donor vector, the 3' end of the left arm is immediately upstream of ATG. So therefore, the GFP will be under the native gene promoter. So in summary, both the gRNA target around gRNA ATG region, and the left arm is immediately upstream of ATG. GFP is under native gene promoter, and your targeting gene is knocked out. So this shows the screenshots of the product page, what the product page look like. So on the top, you will see ATG5. That's the name of the gene, and has nothing to do with the starter codon ATG. Human gene knockout via CRISPR. So here's the SKU, the catalog number, and that's the description. This shows the kit components, 4 components, 2 gRNA vectors, EMP Cas vector, and the donor vector containing the GFP puro cassette. That's the negative control. We also have the kit in different donor vectors. There's a button. If you, for example, if you want to use a different selection marker or a different fluorescence protein, you can click the button and it will show the options. Here you will see another 3 kits for the same gene, but in other donor vectors, either RFP blycidin or luciferase puro or blue BFP neo. So next I will talk about the step-by-step protocol, how to use the CRISPR gene knockout kit. So first, you need to do a cold transfection of the gRNA and a donor vector into cells. There will be 3 separate transfections, each of the gRNA and the scramble control with the donor vector into cells. So the transfection reagent you use depends on your cell line. You need to choose a transfection reagent that gives you a high transfection efficiency. OriGene also offers several transfection reagents. Here's the website. You can take a look if it works for your cell line. So after transfecting into cells, the CRISPR-Cas9 will cut at the location specifically, and then homologous recombination occur. The GFP puro cassette will replace the target gene. And this process only takes about 1 week. So now you are very happy. Oh, it's very fast. However, at this stage, the edited cells, the percentage is low. So the next step, screening the edited cells, that's the most challenging and tedious to fish out the edited cells. Because there is a selection marker integrated into the genome, so we can use a puro to help selection. But there's a question, when to apply puro selection? Can you apply puro selection just 1 week after transfection? The answer is no, because if you do, you might get stuck in the screening process. Okay, so puro-resistant cells. Of course, the edited cells has integrated GFP puro cassette. It will be resistant to puro selection. In addition, the transfected cells, but unedited, it has a donor plasmid as episomal form. In the donor plasmid, because the puro marker is under PGK promoter, which is constitutive. So therefore, even the donor plasmid will provide puro resistance. So therefore, before applying puro selection, we need to decrease the second group. Because the donor plasmid does not replicate during cell division, so we can passage the transfected cells about 2 to 3 weeks to dilute cells containing the donor vector, and then apply pure selection. So this way, you will have a significantly decreased the background. So the diagram shows the edited cells will, with the cell division, of course, the edited chromosome will divide, replicate, and present in daughter cells. But the donor plasmid will be lost in the daughter cells. Similarly, in unedited cells, the donor plasmid will be lost in majority of cells. So in this way, you apply pure method selection, it's easier to get the real knockout cells. So the next step is apply pure selection. So puromycin doses need to be determined by kill curve for each cell line because every cell line is different. You will need to choose the lowest dose that completely kills the untransfected cells after 4 to 7 days of a puromycin selection. If it's necessary, you can isolate the single cell colonies. There are 2 methods. One is the limiting dilution, one is the cloning rings, cylinders. So the limiting dilution is to seed 1 to 2 cells per well. So you can look under the microscope and mark the wells with only 1 cell and let it grow further. The cloning ring cylinder uses a larger dish, such as a 10-centimeter dish, and seed more cells. You do 1 to 50 split, and you can apply puromycin selection at the same time. So pure resistant cells will grow into colonies, and you can apply the cloning rings or cylinders around the area of the colony, watch it, trypsinize, and transfer the cells out to 96 well plate and expand them further. So here shows the diagram of cell passage in the selection. So the passage 1, P1, that's 48 hours post-transfection, and you do a split and grow for 3 more days. That's a passage 2, 5 days post-transfection. So at this stage, the integration already occurred. So you have an optional step to do genomic PCR. So however, at this stage, the edited cells percentage is low. And genomic PCR tend to be difficult because it tends to have a repetitive sequence or GC-rich sequence. So if it's this genomic PCR is inactive, don't worry about it. Still continue passage cells and then apply pure selection. So at this step, we also recommend to freeze or keep raw just in case anything happens, and you can select again. So next step is to analyze the pure positive cells if your gene is knocked out. So one way, if you have a good antibody, you can do Western blot. Western blot, you use a whole cell population. It's better to use after isolating single colonies. And another way is to do genomic PCR. To verify GFP pure integration, then sequence the PCR product to confirm the integration. Genomic PCR, you can design specific primers, only amplify the edited chromosome. So that's the left arm, that's GFP pure cassette, that's the right arm. So this region also present in donor DNA. So therefore, to avoid amplifying the donor DNA, you need to, the primer for the left junction, the forward primer needs to be upstream of the left arm. And reverse primer is in the GFP region. So this way, it only amplifies the edited chromosome, not the donor DNA. So this primer pair is to amplify the right integration junction. So the forward primer is in the pural region. The reverse primer should be downstream of the right arm. Okay, so next I will show you some validation data. Okay, so the kit we use to validate the system is ATG5 knockout. I already showed you this page before. So what we did is we co-transfect the gRNAs with the donor vector and scramble control separately into HEC293 cells and the passage for 5 times then apply pure selection. Here is the pure selection data. So the hectonet base was selected under 1 microgram per ml puromycin for 5 days. So the scramble, the donor cells all died, and both target 1 and target 2 have happy cells, but with the target 1, it has more cells. So suggesting that target 1 is more efficient. So next, we did genomic PCR to verify the integration. So the primary pair, the forward is the upstream of the left arm, and a reverse primer is the GFP region that to amplify the left integration junction. The epi count is about 920 base pairs. Here shows the data. So actually, the genomic DNA we use, that's before purification. So we are able to get genomic PCR to work. And so even before pure selection, we know that genome editing worked. So the scramble control is negative, and both targets generate correct size of PCR products. And with the target 1, there's more PCR products, which is consistent with the pure positive cells. So next, we sequence the PCR products to verify everything is correctly integrated. So here the sequencing data. This sequencing data use the forward primer to do sequencing to verify the 5' end of the left arm is integrated into genome if it's correctly integrated. So this is top line, that's the genomic sequence. And the bottom line, that's the sequencing data from the PCR, which is from the edited chromosome. So you will see that it's exactly like a genomic sequence. So the 5' end of the left arm is integrated into the genome correctly. And this is the chromatogram showing the sequencing peaks. Next, we want to know if GFP is correctly integrated. We use the reverse primer to do the sequencing. So that's genomic sequence, and here shows the coding region of ATG5, and that's the sequence from edited chromosome. Before the coding region is exactly the same as the genomic sequence, and follows with a 6 base pair. That's the cloning sites we added before GFP, and it follows with the GFP sequence. So this is showing the GFP is also correctly integrated. Here's a summary. CRISPR, the knockout kit, that's a completely gene knockout kit. We have a knockout kit, it's a locus specific. We have a specific kit for every gene. So it will have specifically the gRNAs for each gene, 2 gRNA vectors. Donor vector also specific for your gene, has the specific homologous sequences and the control. So it's ready to use. You receive it directly, transfer into your cells. The target gene will be knocked out, and will knock in a GFP pure cassette to help selection. So now I have shown you such a wonderful kit, how to find the gene knockout kit on the website. This is the OriGene's landing page. There's a search box on the top. You can search with the gene symbol. I use TP53 as an example, and click search. This is a search result page. All the products related to TP53 are categorized, either antibody or protein, CDN clones, RNA, qPCR. This is the CRISPR tab. So click on the tab. It will show you 2 products, one for human, one for mouse. If you are interested in human, just click on the link. That's the page I already showed you, the product page. Okay, for customers who are interested in the general information or other videos, we have CRISPR resources. All the information I present today is in the CRISPR manual. It can be found on the CRISPR landing page. We also have the videos and the webinars, quick learning or gene knockout you already watched before the webinar started, or general information about CRISPRs. So I will be happy to answer any questions.
All right, thank you, Minjuan, for this a nice presentation. As we as instructed at the beginning, And so questions, I can see a lot of people already putting in questions into the question box. We will spend 15 minutes to address some of the questions. Okay. So one of the questions, Minjuan, they ask you is, what is the size in our vector? What is the homologous arms? What is the size we choose?
Okay. The homologous arm, both the left side and the right side, we use 600 base pairs, and it works for us. So all of the knockout kit and the arms, each side is 600 base pairs.
So is 600 base pairs sufficient, you think?
Yes. Some people have tested to use, companies have tested to use 700 or 800, but in our hand, 600 works good too.
Okay, so basically, OriGene has been working with 600 base pairs, and we have validated it works well, and we have also many happy customers that have been using the same design and be able to achieve successful editing. So whether this can be further optimized, of course, I believe in any experiment design that can be optimized based on the insert size, based on the cell line that a different homologous recombination occurrence. So it can be optimized. But the good thing about our kit is we have a pure myosin selection. So it basically can bear, it's robust. You can bear various selection homologous recombination efficiency. So even something that didn't occur at the optimal rate, the successfully edited cells can be selected using pure myosin. So this overcome the issue of various transfection, editing efficiency. Okay, so let's see whether, let's read another question. So there's a customer asking in the knocking down in different organisms, like honeybees and bumblebees. That's an interesting question.
It's very interesting.
You want to answer that?
For those, if the live organisms, maybe use virus, maybe, or I don't know, the embryos, you can use microinjection, gRNAs and Cas9 mRNAs, that might work. If you want to use the lenti system, so the donor vector, it needs to be packaged with a non-integration packaging kit, and OriGene is in the testing that kit. So I believe soon we will have that kit available.
I think so. The living organism, that CRISPR editing has been done in animals all the time, in ES cells, in mouse. That has been done, but sorry for my limited knowledge about bumblebees or honeybees. I don't know whether those insect cells has been used for live cell gene delivery. So, but organism definitely, even in mammalian cells, that has been done. There's another question about the purpose for two guide RNAs. The question say why we include two guide RNAs?
Sure. The reason we offer two guide RNAs because you don't know that if the sequence will cut the DNA efficiently. So that's the reason we offer two. Both the gRNAs target around 80 regions. So you can use one donor, but two different gRNAs. They also be tested separately just to make sure one might work better than the other one. Just in our validation data, we have two different gRNAs. One works better than targeted two.
So basically, having two, we just have a better how to say
chance.
Yeah, it's lower the risk of unsuccessful editing, so you can have a successful rate.
Yeah, if you want to order more gRNAs, we can provide a custom kit for it too.
Oh, this is a good question. I think I have the same question about. You mentioned GFP expression is under the native promoter. Can GFP be used in fact using GFP versus just GFP positive cells to do the selection?
Sure. Yeah, that's a very good question too. Yes, because the GFP is under the native gene promoter, so that totally depends on the strength of your promoter, if it's constitutive or not. If it's strong promoter and it's constitutive promoter, absolutely, you can do FACS sorting just 1 week after the transfection. It's going to be faster. Sort out GFP positive cells. And yeah, so you might not even need the pure selection.
So the short question to that answer is, a short answer to that question is, basically that depends on your promoter, the gene that you're working with, whether that is high expressor or they do express in the particular cell line that you use in this research. So so this is a conditional, depends on the target you study. So.
Okay, I see one question. Shouldn't both the gRNA vector be applied to the same cells and well, otherwise there won't be 2 cuts? Okay. So even with the one gRNA, we'll bring Cas9 to the location. Then Cas9 has 2 domains, which will cut both strands, leading to double-stranded break. So you only need one gRNA to lead to double-stranded break. And then the donor vector has homologous arms, will do homologous recombination, and your gene will be knocked out. And if you still have the questions, you can always contact our technical support team.
Actually, in our standard protocol, we recommend you to use 1 sgRNA with the donor in 1 transfection. However, I think this can be, to have both guide RNA in 1 co-transfection should still work, but it's not required. Because the guide RNA, the concept is you introduce a double-stranded break. The double-strand break can be introduced by 1 enzyme, 1 guide RNA, or it can be introduced by 2 guide RNA. In this case, then you have a middle piece removed. But both doesn't matter whether single cut or double cut. But it definitely have to cause double-stranded break, and then the homologous recombination will occur. During the repair process, the RNAs, the genomic DNA is getting edited. So we don't have to have 2 guide RNA, but we don't mind having 2 guide RNA in the same tube as well. Go next one.
Okay. So there's a question, if can CRISPR edit genes on plasmid instead of chromosome genes? The answer is yes, it can. If you design gRNA cutting a specific plasmid and you do co-transfect into your cells, it should work too. It's not just cutting the chromosome DNA, it cuts any plasmids. Some questions are specific. You can we'll try to answer questions applied to most customers. One question is, what is the percentage of like a heterozygous knockout cells with this process, or how you can get biallelic or monoallelic knockout? That's a very good question. Homologous recombination, most of the cells, you will get the cassette knocking in probably 1 allele. However, the CRISPR system is very efficient, so the other allele might still be targeted by the CRISPR. The indels might occur. So that's why you can do genomic PCR. You can amplify to see if you have endogenous allele or not, and do sequencing to confirm the other allele if you have indels, insertion, or deletions. That information is also in the CRISPR manual, so maybe it's easier to digest.
Yeah, so there's a question related to that. How do you assay if this when you get the selected puromycin resistant cell, whether they are biallelic knockout or single allelic knockout.
Okay, yeah, just as I mentioned that you can design genomic PCR primer to amplify edited chromosome and design primer to amplify the endogenous allele. So if you get products for both, that means your the knockout is monoallelic. But you need to sequence the endogenous allele to make sure that it does not have indel. If it does have indel, perfect. You have both knockout. If it does not have the indels, you can do another targeting with a donor vector with a different selection marker. So you can select again. That information is also on the manual. So
here is a question. Say now you have shown your expertise that in mammalian cells knockout, but I work in plants. But how can you help me? Either does OriGene have any intention of develop plant-based CRISPR tools?
Yeah, current OriGene doesn't have a CRISPR for plants.
Yeah, right now our expertise indeed is limited to mammalian cells, but we would love to work with you, the plant scientist. Maybe throughout the process, we can also learn something from you. How does the plant gene delivery work? And we'd love to create join force with you. And so please email us when people that work on plants or working with different system other than mammalian cells. We'd love to work out the tools that is suitable for your field. Is this kit used in mice?
This kit used in mice. With the mice cell line, might be it should, but with the making knockout mice in using kit, people tend to do microinjection of the gRNA and mRNA into the yeast cells or embryos.
So for knockout mouse, most people use using RNA microinjection. We have tools for that. Basically, you have to work with ES cells to engineer the ES cells, then implant that one into pregnant mouse. How do you perform a single cell selection? I thought we went through that.
Yeah, I think we did that. There are two ways. If before pure selection, you can use the standard cloning rinse, and after pure selection, you can use the dilution method to seed when the two cells per well.
Yeah, basically that general question about single cell cloning. Yeah, was using cloning ring or the two mammalian cells.
So one question is that could you say a few words about off-target? So that is the question people always ask. So the off-targeting, that means if there's another sequence that the gRNA CRISPR recognize, it will cut it. If that sequence does not have the PAM motif at 3' end of the target sequence, it will not be targeted at all. And the 10 bases at the 3' end of the target sequence, that's called seed region. If there are mismatches in that region, the targeting efficiency probably is almost to 0. So when we design gRNAs, we consider those off-targeting issues too. If you are really concerned, we have a Niki's vector. But for most research projects, when you design gRNA using those strategies, it should be okay.
I think due to time limit, we have a long list of questions. It was impossible for us to address them one by one. I think it's the time constraint. We will just answer the very last question. For the rest of them, we will have email follow up with you. So, Minjun, let me see whether I can pick one question. Maybe have a general appeal. Okay. Can you knock out 2 separate genes in one experiment?
2 separate genes. Okay. Using the knockout kit, because the donor vector is kind of big, I don't recommend. For indels, it's doable. Just transfect 2 gRNAs into your cells. It's doable to knock out 2 genes at the same time.
Yeah, so here I just want to clarify the knockouts, the word knockout, gene knockout, they have 2 different types of knockouts. One is just using CRISPR to introduce indels, and due to error-prone repair, then causing the off-target,
introduce variable insertions,
deletion, therefore turn off the gene. So that is that is a non-templated knockout. But the other one is using homologous recombination to insert a cassette to enable your selection. So OriGene's knockout kits is a second type of knockout, which will need a template and introduce a selection marker to enable the downstream enrichment of the edited cells. So for double knockouts, it can be performed using the first form, which is indel-based knockout. That can be done by basically introducing multiple guide RNA into the cells. That has been achieved in cells. They can knock out multiple, not just 2, 3, 4, indel-based knockouts. But for the templated form, so we need a co-transfection of the template, and also need a homologous recombination. That is a lower efficiency and lower occurrence, the percentage. So for that to have double knockout, that is very low frequency. So we recommend it for the templated knockouts, do 1 gene at a time. You can do that serially. In the sense, you can do 1 knockout, 1 gene, and then flux out the puromycin, and then you can use the second kit to knock out the second gene and continue using puromycin selection. So that's for double knockouts. Okay, I really appreciate that many of you stay online for so long, and we will wrap up today's webinar now, and we will follow up with you and with the presentation as well as the recording. So if you have any other questions, you can always address our tech support. And for those who already posted the question, we will answer them individually to you. Thank you so very much.
Thank you.
And have a great day, a great